Here we focus on cellular acid-base regulation. It's a topic that really shouldn't be overlooked because the body has about 40 trillion cells in it. Our figure shows a representative cell, and in that cell we have a box that says variable metabolic activity. That is simply a reminder that there are numerous metabolic reactions that occur in cells, and many of them produce acids. Now, the figure does show CO2 being produced in cell metabolism, and that's specifically cell respiration and the Krebs cycle. CO2, of course, is not an acid, but in the body fluids, it becomes hydrated to make an acid, carbonic acid. Now, as cell metabolism changes proton concentrations in the ICF, we have three different kinds of buffering systems poised, ready to dampen those changes. They include physiochemical buffering, that's chemical buffering by the phosphate and protein system, metabolic buffering, alterations in enzymatic function that modify metabolic rates to actually adjust the rate at which protons are released, and organellar buffering, the ability of some organelles like lysosomes and mitochondria to sequester protons and thereby change their concentrations in the ICF. After all this is done collectively, that is, acid production, buffering, and so on, we end up with an ICF pH that ranges between 6.8 and 7.1. Compared with the ECF pH, which is generally about 7.4, the ICF is more acidic. That would cause us to assume that protons would have a tendency to leak or diffuse out of the cell into the ECF. But that really doesn't happen because we also need to consider the transmembrane voltage, which is inside negative, outside positive, which has a tendency to attract protons in from the extracellular fluids. So it's really the entire electrochemical gradient that needs to be considered. These cells are steadily acidified because of proton movement down that gradient. We have another ion which is subject to moving down its electrochemical gradient, and that's the bicarbonate ion. I'm mentioning that here because movement of bicarbonate out of the cell has the same effect on acid-base balance in the cell as a proton moving in. The reason for this is that bicarbonate is, of course, a base anion, and it has a tendency to bind to a proton. And so as that occurs, the bicarbonate binding to the proton helps to keep the proton level low. But if we're losing bicarbonate from the ICF, its effect basically is the same as a proton moving in. Logic tells us that if this passive acidification is happening basically all the time, cell membranes must have transporters in them that serve to act as either acid extruders, shown on the right, or acid loaders, shown over here on the left. Let's think about the acid extruding mechanisms first. How would we do that? Well, one way is to pump protons out of the cell. The other way is to mediate the transport of base anions, like bicarbonate, into the cell. Like I said, both of those basically have the same effect. So they extrude acids. It could be, under some circumstances, that we need to do the opposite. That's to load acids into the cell. And of course, we could do that by having transporters that pump protons in and or base anions out. Let's assume that after the cell's done its metabolic activities and buffering and transmembrane transport of protons and base anions, suppose we have a change in ECF pH. What then happens? Well, the ECF has numerous buffering mechanisms in it that will dampen those pH changes. Also, the blood is part of the extracellular fluids, and the blood being carried to the lungs and kidneys, those organs will make final corrections in the pH. And specifically, the lungs do it by controlling the rate of CO2 elimination, and the kidneys can do it by producing either, as necessary, an acidic or alkaline urine.